Welcome back for another deep dive. This time we're going to be taking a look at a collection of scientific papers that span a really impressive range of topics. We've got nuclear reactions, accidental chemistry breakthroughs, and even early stem cell research. I'm excited to see what we find.
It really is amazing to me how fields that seem completely unrelated can end up intersecting in really interesting ways. I think we're in for some surprises today.
So let's get started with something that's always fascinated humans, and that is, of course, stars.
Always a great place to start.
This excerpt we have here from NBL 399.pdf goes right to the heart of how stars create their energy. It talks about the proton reaction, and that's the process that fuels our own sun.
Right, and it all starts with hydrogen nuclei. I mean, they're basically just protons. And under incredible heat and pressure, they slam together and fuse to form helium.
Wow.
And that releases a tremendous amount of energy. That's what makes stars shine.
Incredible. So our sun is really just a giant nuclear fusion reactor in space.
That's one way to think about it.
But the source also mentions that this proton reaction isn't actually powerful enough to explain the energy output of bigger stars.
Yeah, that's right. In those massive stars, the temperatures and pressures are even more extreme, and that allows heavier nuclei like helium to start fusing as well.
So is it like a chain reaction?
Yeah, you could think of it that way. The fusion of lighter elements sort of creates the conditions for heavier elements to fuse. The source even talks about the formation of beryllium-8, which is unstable. But it's actually a crucial step in the process because it leads to the creation of carbon-12.
So it's like a cosmic building process.
That's a great way to put it.
Amazing. Okay, well, now we're going to shift gears a bit from the vastness of space and go into a laboratory setting. This excerpt from NBL 400.pdf is a first-hand account by a scientist named A.J.P. Martin. He played a big part in developing partition chromatography. But it seems like he made a surprising discovery in his early experiments.
He did. And it really goes to show how sometimes accidental discoveries can lead to big breakthroughs. It turns out that impurities in his chloroform, specifically alcohol, ended up playing a role in separating the compounds he was working with.
Wait, so impurities actually helped?
That's right.
Yeah.
It's funny because normally you'd want everything to be as pure as possible, but sometimes those imperfections can lead to something new and unexpected.
That's a great point.
Martin himself actually said that taking less care, at least in those initial experiments, could actually be more productive because it lets you learn from unexpected results, even failures.
That makes a lot of sense. So you don't always win by getting everything perfect. It's good to remember that in science, the path to discovery isn't always straightforward.
Sometimes it's those twists and turns that lead to the most interesting findings.
Absolutely. Now let's move on to NBL 401.pdf, which is a Nobel lecture by a particle physicist named Toshihide Masukawa. He talks about his journey to try to understand elementary particles, which are like the fundamental building blocks of matter. And he mentions being fascinated by the work of another physicist named Nambu, who is studying this concept called spontaneously broken chiral symmetry. Could you maybe help unpack that a little bit for our listener?
Okay, so imagine a symmetrical object, like a perfect sphere.
Okay, I can picture that.
Now imagine gently squeezing it, so you're breaking that perfect symmetry. Well, that's sort of what happens in particle physics. Chiral symmetry basically refers to a kind of balance between certain particles.
Okay.
But sometimes this symmetry gets spontaneously broken, and this can have huge implications for how these particles behave.
Wow, so the laws of physics themselves can be a little wonky.
It definitely seems that way sometimes.
So how does NBL 402.pdf connect to this particle physics idea? Because on the surface, it seems kind of unrelated. It talks about comparing the spectra of X-rays to the spectra of alkali metals.
That's a great question. It is a bit of a jump, but it all comes down to the behavior of electrons within atoms.
Oh, okay.
Both X-rays and the light emitted by alkali metals involve these transitions in energy levels of electrons.
Right, so it's like electrons jumping up and down energy level.
Exactly. And when an electron jumps between those levels, it can absorb or emit light. And the pattern of that light creates a kind of fingerprint for each element.
Oh, I see. So by comparing these fingerprints, you can see similarities in how electrons behave even in different elements.
That's it. It's like uncovering a hidden code in the language of the elements.
That's really cool. Speaking of codes, let's move on to NBL 403.pdf. This is a Nobel lecture by Hannes Elfven. He's a pioneer in plasma physics. Now, could you remind everyone what plasma is and why it's so important?
So plasma is sometimes called the fourth state of matter after solids, liquids, and gases.
Okay.
Basically, it's a gas that's been superheated to the point where atoms start losing their electrons. And that creates this mix of charged particles. And the crazy thing is that something like 99% of all visible matter in the universe is plasma.
Really?
Yeah, stars, nebulae, even lightning.
Wow, that's amazing. So what did Elfven focus on in his research?
He looked at different ways to study plasmas. He contrasted the hands-on, messy experiments like you'd have with electrical discharges with the elegant mathematical theories that come from studying normal gases.
So, two very different approaches. Which is better?
Well, Elfven actually argued that both are essential. He even talked about this problem in nuclear fusion research called the thermonuclear crisis.
Okay.
And basically, it was a situation where the theoretical predictions didn't match what they were seeing in experiments at all. So it showed that relying too heavily on either theory or just experiments can lead you down the wrong path.
It's about finding that balance.
Yeah.
All right, well, let's switch gears again and dive into the world of stem cells. NBL 404.pdf is a Nobel lecture by Martin Evans, who's a pioneer in that field. Now, he faced a big challenge early on: how to maintain something called pluripotency in stem cells when they were grown in a culture. So could you explain what pluripotency is and why it was so hard to achieve?
So pluripotency is this amazing ability that some cells have, particularly embryonic stem cells, to become any type of cell in the body.
So it has the potential to be anything.
That's it. But trying to keep that flexibility outside the body, like in a petri dish, was really tough. They tended to specialize and just become one type of cell.
So how did Evans figure out how to overcome this?
He had this really important insight that these differentiated cells, the ones that had already specialized, could actually help support the pluripotency of other stem cells.
Oh, that's interesting.
So they need their more mature siblings to stay flexible.
Even at the microscopic level, community is important.
You can say that.
Yeah.
And Evans actually predicted back in 1975 that we'd be able to culture these pluripotent cells directly from embryos.
Wow.
And, that actually became a reality. He also talks about things like retroviral vectors and homologous recombination in his lecture.
Oh, right. Those are pretty high-tech ways to manipulate genes.
Exactly. They're essential tools in genetics research these days.
Well, he sounds like a true visionary in this field.
Okay, let's change gears again and dive into organic chemistry with NBL 405.pdf. This features a lecture by Otto Wallach, who is fascinated by these compounds called terpenes.
Yeah, terpenes are everywhere in nature. They're what give a lot of plants their characteristic scents, like that piney smell in a forest or a citrusy aroma from an orange.
Oh, right.
And people use them all the time in perfumes and essential oils and even some medicines. But back then, their structure was a complete mystery. Wallach even described it as being hidden behind a veil.
Wow, so how did he figure it out? With a lot of experimentation. He was a master of organic chemistry, and he was able to determine the structure of all kinds of terpenes.
And that knowledge was really important.
Extremely important, because it meant scientists could start synthesizing these compounds in the lab. And that's had a huge impact on industries, from fragrances to pharmaceuticals.
Amazing. Okay, now let's explore NBL 406.pdf. This is a lecture by Cyril Hinchelwood, and he was really interested in something called chemical kinetics.
Chemical kinetics is basically studying how fast reactions happen and what affects their speed. It's kind of like studying the traffic patterns of molecules as they bump into each other, react, and form new substances.
Like a molecular highway. So what reactions did he study?
He focused on ones that seemed pretty simple on the surface, like combining hydrogen and oxygen to form water. But as he dug deeper, he realized they were way more complex than anyone thought.
What made them so complex?
The discovery of chain reactions.
Right, where one reaction sets off a whole cascade of others.
Exactly. So these seemingly simple reactions weren't just happening in one neat step. It was more like a domino effect at the molecular level. And he even talked about using nitric oxide to study these chain reactions, because it has this ability to interrupt or slow them down so you can figure out which steps are actually part of the chain.
It's like a molecular detective story.
In a way, it is.
Okay, well, I think we need to take a slight detour here and talk about NBL 407.pdf. It's all about bile acids and the research of Heinrich Wieland. He described these molecules as really large and kind of monotonous because they're just made up of carbon, hydrogen, and oxygen.
He wasn't wrong. But figuring out the structure of these molecules was a real puzzle. They're so big and complex.
So how did he do it?
Well, he had to use some clever chemical tricks, like decomposition and oxidation. It was like he was taking apart this giant Lego structure brick by brick to see how it was built.
So he's like a molecular detective, too.
Definitely.
Okay, now back to physics.
Back to physics it is. So NBL 408.pdf is Charles Barklay's Nobel lecture on X-rays, a pretty powerful tool for figuring out the structure of atoms.
X-rays, yeah. The things that can see through our bodies. What did Barkla discover about them?
He made this really important connection between X-ray emission and the way electrons behave. He found that when you hit an atom with energy and knock out an electron, it releases a very specific X-ray.
Okay.
It's like each element has its own unique X-ray fingerprint.
Wow, that's incredible.
Yeah.
We're basically listening to the conversations between atoms and energy. This discovery really leads us right into quantum physics.
It does, and that takes us perfectly to NBL 409.pdf. It's Robert Millikan's lecture, and it focuses on his work on the electron and this phenomenon called the photoelectric effect.
Ah, Millikan, the oil drop experiment guy.
Exactly. That experiment was brilliant. He was able to actually measure the charge of a single electron.
That's amazing when you think about how tiny an electron is. What about this photoelectric effect, though?
So that's when you shine light on a material, and it can actually knock electrons loose.
Oh, okay.
And Millikan's experiments ended up being the first direct proof of Einstein's photoelectric equation.
Why was that such a big deal?
It was a huge victory for quantum theory because it showed that light wasn't just a wave, it also acts like these little packets of energy called photons. It completely changed how we understand light and matter.
So he's saying that light can be both a wave and a particle at the same time.
That's exactly it.
That's mind-blowing. So how does MBL 410.pdf build on this quantum idea?
This lecture dives into the weird world of superconductivity. That's where electrical resistance completely vanishes.
Really? It just disappears.
Yep. The lecture is by John Robert Schrieffer, and he and two other scientists, Bardeen and Cooper, developed a theory to explain it.
It sounds like something out of science fiction.
It does, right. But it's completely real. And their theory basically says that at super low temperatures, electrons can actually pair up and form these things called Cooper pairs.
So they buddy up.
That's a good way to put it. And when they're paired up, they can move through the material without any resistance at all.
It's like they've found a secret passage.
I like that. A secret passage through the world of physics.
Okay, my brain is definitely starting to get a little overloaded with all this quantum stuff.
I hear you.
Can we maybe take a break from physics for a minute and explore something a little more biological?
Sure, let's do that. NBL 411.pdf features Peter Medawur's Nobel lecture, and it's all about his explorations of the immune system.
Okay, the immune system.
He did some really interesting experiments with chicken embryos. He was focusing on leukocytes, which are those white blood cells that protect us from invaders.
Right.
He was really interested in how our bodies recognize those invaders.
Okay, so he was looking at the first line of defense.
That's right. And in his experiments, he would trigger an immune response in chicken embryos. And he noticed that these leukocytes would cluster around the infection site, forming what he called foci.
So it's like a battleground for the immune system.
Exactly. And these foci weren't just static, they were actually areas where cells were rapidly dividing and multiplying.
So the body is essentially mobilizing its troops.
That's a great way to think about it.
That's so cool. But what's this about a connection to evolution?
Okay, so this is where it gets interesting. Medawar suggests that the way our bodies recognize and fight off foreign invaders might be linked to how cells differentiate and form tissues during development.
So from fighting germs to actually building a body.
That's the idea.
Wow, that's a pretty deep connection. Okay, let's shift gears again. NBL 412.pdf is all about Nikolai Semenov and his work on combustion.
Semenov really changed how we think about fire. He challenged the old idea that combustion was just about things spontaneously bursting into flames. Instead, he showed that chain reactions are really important.
Right. Those chain reactions keep popping up everywhere.
They really do. And in a fire, you have these really reactive molecules called radicals, which are basically like tiny fire starters. Semenov actually showed that radicals are present during combustion, and they're key to keeping those chain reactions going and the fire burning.
Wow. It's amazing to think about all those tiny reactions happening so fast to create something as powerful as fire.
It really is.
Okay, let's switch from the destructive power of fire to something a bit more constructive. What can you tell us about NBL 417.pdf and Fritz Haber?
This lecture focuses on a really important discovery: how to synthesize ammonia from its elements.
Ammonia, like the stuff in cleaning products.
That's the one. Back in the early 1900s, there was a big need for nitrogen fertilizers, to feed a growing population.
Right, but nitrogen is a gas, right?
Exactly. And getting it out of the air and into a form that plants could use was a huge challenge. Haber figured out how to combine nitrogen from the air with hydrogen to create ammonia.
Wow, that sounds like a really tough reaction to pull off.
It was. It required incredibly high pressures and temperatures, plus some special catalysts. But his breakthrough really revolutionized agriculture.
What a game changer.
Yeah. It's estimated that over half the nitrogen in our bodies comes from the Haber-Bosch process, which is the industrial process he helped develop.
From the smallest atoms to the biggest challenges, this deep dive is really covering it all. Now let's talk about NBL 418.pdf, which features Albert Szent-Györgyi and his work on biological oxidation.
Szent-Györgyi was interested in how living things use hydrogen as their primary fuel source.
Wait, hydrogen as fuel, like in a car?
Not quite like that.
Right.
But think about it, our bodies are basically machines that run on hydrogen. We break down food, extract hydrogen atoms, and that's what powers all those processes that keep us alive.
So we're all hydrogen powered.
We are.
Wow. So what else did he discover?
Well, he also talks about this early work he did on the browning of plants, like when you cut open an apple and it starts to turn brown.
Right.
And this seemingly simple observation led to a pretty big discovery.
Really?
Yeah, he stumbled across this substance he called hexuronic acid, which turned out to be vitamin C.
No way. Another accidental discovery.
It really goes to show that sometimes the most important breakthroughs come from those unexpected observations.
That's so true. Okay, now let's talk about Paul Lauterbur and his work on magnetic resonance imaging, or MRI. That's NBL 419.pdf.
MRI is an amazing technology. Doctors can see inside the body without having to do surgery. And Lauterbur started with these really simple experiments where he used gradients in a magnetic field to create images.
Okay.
He was basically looking at how nuclear spins were distributed.
Nuclear spins, can you explain that?
Okay, so different atomic nuclei act differently in magnetic fields.
Okay.
And by looking at how they respond, you can create an image of how they're distributed within a sample.
So it's like mapping the invisible world inside us.
That's a great way to put it.
And what's really amazing is how fast MRI went from being a research tool to something that's used in hospitals every day.
It is. It shows just how powerful scientific ingenuity can be.
All right, next up is Edwin McMillan and NBL 420.pdf, which is about transuranic elements.
Okay, so those are elements that are heavier than uranium on the periodic table. McMillan actually discovered the first transuranic element, neptunium, and his story is kind of like a detective story.
Oh, I like a good mystery. Tell me more.
He was studying uranium fission products, which are what's left over after you split a uranium atom.
Okay.
And he noticed something weird, this radioactive activity that lasted for about 2.3 days. At first, he thought it was a rare earth element.
So he thought he knew what it was.
Right. But as he did more experiments, he realized it was behaving really differently from other fission products. And that's when he realized he had found a brand new element.
Wow, what a discovery. It's like finding a needle in a haystack.
Absolutely. It's a great reminder that sometimes the most groundbreaking discoveries are hidden in plain sight.
Now, what about NBL 421.pdf? That looks like it's about Arthur Hardin and alcoholic fermentation.
That's right. Alcoholic fermentation is what makes bread rise and gives beer and wine their alcohol content.
It's a pretty important process.
For sure. And Hardin's big discovery was that phosphate plays a key role in this process.
Really? How so?
Well, during fermentation, this compound called a sugar phosphate forms.
Okay.
And this showed that phosphate is an essential ingredient in the whole process. He even made a connection between phosphate's role in fermentation and its potential importance in forming bones.
Wow, what a connection.
Yeah.
Now, let's switch gears completely and talk about Paul Krugman and his work on international trade patterns. That's NBL 422.pdf, right?
That's the one. He was trying to explain why countries that are really similar in terms of their resources and tech still trade with each other.
That seems counterintuitive.
It does, but Krugman showed that even similar countries can benefit from specializing in slightly different products and then trading. It makes things more efficient, and it gives consumers more choices.
So everybody wins. Okay, now back to the microscopic world with NBL 426.pdf and Salvador Luria. He studied bacteriophages and bacterial genetics.
Bacteriophages are those viruses that infect bacteria.
Like little assassins.
Exactly. Luria was initially interested in how these phages could change the surface of Salmonella bacteria. But this led him to a broader interest in bacterial membranes, which are basically the gatekeepers of the cell.
Right. They control what goes in and out.
Exactly.
So what did he focus on in his later work?
He turned his attention to collicins, which are toxins that some bacteria make to kill off their competitors.
So it's a bacterial arms race.
You could say that. And Luria realized that studying collicins was a way to understand how bacterial cells are organized.
So it's like using these tiny weapons to unlock the secrets of the cell.
That's a great way to think about it.
Okay, well, for our last stop on this deep dive, let's talk about Carl Landsteiner and his discovery of blood groups. That's NBL4twinheaven.pdf.
Right. That's right. This was a groundbreaking discovery that made blood transfusions safe. Before Landsteiner, blood transfusions were incredibly risky because nobody knew about different blood types.
So he realized that not all blood is the same.
Exactly. He figured out the ABO blood group system. Before that, people were mixing incompatible blood types, which could trigger a really dangerous immune response.
It's hard to imagine modern medicine without this discovery.
It's a real testament to how a single scientific breakthrough can have a huge impact on human health.
Okay, I think that's a good stopping point for now. This first part of our deep dive has taken us all over the map, literally.
It really has. We've explored stars, accidental discoveries in chemistry, the complexities of cells and molecules, and so much more.
And this is just the beginning. I can't wait to see what awaits us in the second part of our deep dive.
Me too. Ready for a little cosmic journey? We're going to be heading from the heart of our galaxy to the edge of the observable universe today.
I am so ready. I love how these scientific papers are connecting these massive ideas in space with the tiny details of life right here on Earth. It's fascinating.
Absolutely. Well, let's jump into NBL428.pdf, which is Arno Penzias's Nobel lecture. He talks about this incredible discovery of cosmic microwave background radiation. And that discovery completely revolutionized how we think about the very beginning of the universe.
Yeah, like the echo of the Big Bang, right? That faint radiation that's just kind of everywhere in space.
That's it. But to really appreciate it, we got to rewind a little and talk about the early ideas about how elements formed. Scientists used to think that all elements were created inside stars. You know, like those giant nuclear furnaces we talked about earlier.
Right, those fusion reactions where lighter elements combine to make heavier ones.
Exactly. But there were some things they couldn't explain with just stellar nucleosynthesis, like this thing called the mass-4 barrier.
Okay, I'm not familiar with that. What is it?
It basically describes how hard it is to build elements heavier than helium inside stars.
Why is that so hard?
It has to do with nuclear physics.
Mm-hmm.
To make heavier elements, you got to fuse lighter nuclei together. But fusing a helium nucleus, which has a mass of 4, with another nucleus, you have to overcome this incredibly strong repulsive force.
So it's like a cosmic roadblock.
That's a good way to think about it. It limits what kinds of elements can easily form inside stars, but we know heavier elements exist. So that's where the Big Bang comes in.
Okay, how so?
Scientists started thinking maybe some elements were actually formed in those crazy conditions right after the Big Bang, when the universe was super hot and dense.
Like a giant cosmic pressure cooker.
Exactly. And the discovery of cosmic microwave background radiation really supported the idea that the universe started out that way, super hot and dense.
It's amazing to think about. Okay, well, let's switch gears for a minute and check out NBL429.pdf, which is Alfred Hershey's lecture on his work with bacteriophages.
Ah, yes, bacteriophages, those viruses that infect bacteria.
Right. Can you remind everyone what Hershey found out about them?
So he made this really cool discovery about the structure of the DNA and a phage called T2. He found that all of its DNA was a single, long molecule. He called it a unimolecular chromosome.
Wow, so even those tiny viruses have an organized chromosome.
They do. And this was actually a pretty big deal because back then, a lot of scientists thought DNA was a really simple molecule. Hershey showed that even a tiny virus could have a complex genetic structure.
Really interesting. Okay, let's move on to NBL430.pdf, which is Linus Pauling's Nobel lecture. He talks about the nature of the chemical bond. Can you explain what he meant by resonance? It's a concept that was pretty revolutionary.
Yeah, resonance is this idea that sometimes you can't really capture what a molecule looks like with a single static picture.
Okay.
The true structure is kind of a blend of multiple forms.
Can you give us an analogy? I'm having trouble picturing it.
Okay, so imagine a mule. It's not quite a horse, and it's not quite a donkey, but it has characteristics of both.
Right.
Resonance is kind of like that. The real structure of the molecule is a blend of these different forms, and that blend is actually more stable than any of the individual forms.
That's a really helpful analogy. So it's like the molecule is constantly shifting between those forms.
Exactly. And this idea is especially important when you're talking about things like aromatic compounds.
Right. Okay, well, we've covered a lot of ground so far. Let's talk about NBL432.pdf now, which is Walther Nernst's lecture. He was a big name in thermodynamics. Could you remind everyone what thermodynamics is all about?
Thermodynamics is the study of heat and energy and how they relate to work and other stuff.
So pretty important stuff. What was Nernst's big contribution?
He developed something called the heat theorem. It's this really important principle that connects the change in heat during a process to the change in entropy.
Entropy.
Yeah, that's a measure of disorder or randomness.
Got it. Okay, well, let's talk about Otto Diels and his work on the structure of steroids. That's NBL433.pdf.
So steroids are a huge group of molecules. They have this characteristic four-ring structure, and they're really important in biology. They act as hormones, vitamins, even structural parts of cell membranes.
Right, like cholesterol.
Exactly, cholesterol is a steroid. Diels was especially interested in figuring out the structure of cholesterol, and that was a super tough challenge back then. These molecules are big and complex.
How did he figure it out?
He used this clever strategy where he broke the cholesterol molecule down into smaller pieces. He used a process called dehydration to remove water molecules and create those smaller fragments. And then he could study the structure of those pieces and put them together like a puzzle.
That's really cool. Okay, well, now I think we need to take a trip back in time to the early days of atomic physics. Let's talk about J.J. Thomson's Nobel lecture, NBL434.pdf. He was the one who discovered the electron, right?
He was. And that discovery totally changed how we understand atoms.
Yeah.
Before Thomson, people thought atoms were the smallest possible thing.
Right, the smallest building blocks of matter.
Exactly. But Thomson showed that atoms were actually made up of even tinier particles.
So he was looking deep inside matter. How did he do that?
He used these things called cathode rays, which are basically beams of electrons, and he used magnetic and electric fields to bend those beams.
Why would he want to bend the beams?
Because by measuring how much they bent, he could calculate the charge-to-mass ratio of the particles in those rays.
The charge-to-mass ratio.
It tells you the relationship between the electrical charge of a particle and how heavy it is. And what Thomson found was that these particles had a really high charge-to-mass ratio, which meant they were much smaller and lighter than anyone thought.
That's amazing. Okay, let's move on to Lord Rayleigh and NBL435.pdf. He focused on the densities of gases. What did he find?
Rayleigh was known for being incredibly precise in his measurements. He was comparing the density of nitrogen that he got from two sources,
Yeah.
air, and from chemical reaction in the lab, and he noticed this tiny difference between them.
So something was a little off?
That's right. He suspected there might be another gas hiding in the air, mixed in with the nitrogen.
Wow, so a new element.
Exactly. He teamed up with another scientist, William Ramsay, and together they were able to isolate and identify this new gas. They called it argon.
That's incredible. He found a whole new element because he was so careful with his measurements.
And that's why it's so important to pay attention to even the smallest details in science. You never know what they might lead to.
Okay, let's talk about Fritz Zernike and his invention of the phase contrast microscope. That's NBL437.pdf.
The phase contrast microscope was revolutionary. It let scientists see things that were basically invisible under a regular microscope. You know, things like tiny cells.
Yeah, because they're transparent.
Exactly. Zernike realized that although our eyes can't see these tiny phase shifts in light when it goes through a transparent object, he could create a way to make them visible.
So he was making the invisible visible. How did he do it?
He put a special phase plate in the microscope, and that plate would shift the phase of the light that went through the object, and then that would create interference patterns which would make the object appear brighter or darker.
That's so clever. Okay, let's move on to NBL439.pdf, which is Otto Stern's lecture about his work with molecular beams. First of all, what is a molecular beam?
Imagine a really tiny focused stream of atoms or molecules.
Okay.
That's a molecular beam. And Stern figured out how to create them using a vacuum chamber. He could basically fire these beams of particles at a target to study how they behave.
Like a tiny shooting range for atoms. So what did he find out?
One of his biggest discoveries was something called space quantization.
Okay, what is that?
It means that the angular momentum of atoms, so basically how much they're spinning, can only exist in specific, discrete values.
So there are only certain speeds they can spin at.
You got it. It's a really fundamental concept in quantum mechanics.
Wow. Okay, we're in the home stretch now. 2 more Nobel lectures to go. Let's start with NBL 440.pdf, which is Pierre Curie's lecture about the discovery of radioactivity. That was the start of a whole new era in physics.
Absolutely. Radioactivity is when an unstable atom spontaneously emits radiation from its nucleus.
And it can change one element into another.
That's right. Pierre Curie and his wife, Marie Curie, were pioneers in studying this phenomenon. They even discovered new radioactive elements, like polonium and radium.
But their work was pretty dangerous, right?
It was groundbreaking, but it also came with risks. They were working with the materials that they didn't fully understand, and unfortunately, Marie Curie suffered from radiation sickness because of it.
Their story is a reminder of the dedication and sacrifices that scientists make. Okay, last stop. NBL 441.pdf. Alfred Rohner's lecture about coordination chemistry.
Coordination chemistry is all about metal complexes.
Okay, help me out here. What are metal complexes?
Imagine a metal atom surrounded by a bunch of other molecules or ions.
Yeah.
Those molecules or ions are called ligands, and they're bonded to the metal atom in a specific arrangement.
Like a little cluster of atoms.
Exactly. And Werner figured out that the arrangement of those ligands is really important for understanding the properties of the complex.
So he was like a molecular architect.
Yes, he was. He really changed how we think about the structure and bonding in these metal complexes.
Okay, I think my brain is officially full for the day. This has been an amazing journey.
It really has. We've covered so much ground, from the vastness of space to the tiniest details of molecules.
It's incredible to think about the power of human curiosity and the drive to understand the world around us.
And we're not done yet. We've got one more part in our deep dive to go, with even more fascinating Nobel lectures to explore.
Lead the way. Welcome back to our deep dive. It's amazing to me how these Nobel lectures we've been looking at all tie together this tapestry of scientific discovery. I'm excited for this last set.
These last 4 are really interesting. They get at some pretty big questions like the nature of matter, the origins of life, and even how our bodies work at a really basic level.
Sounds like a good way to wrap things up. Let's start with NBL 442.pdf, which features Paul Dirac's Nobel lecture. He takes us into some pretty theoretical territory here, talking about antimatter.
Antimatter. You know, a lot of people think it's pure science fiction, but it's a very real concept in physics.
Right.
So Dirac's work was all theoretical, but it really changed how we understand the universe. He was trying to come up with this equation, a relativistic wave equation, for the electron.
Okay, that sounds pretty complicated.
It was basically trying to combine quantum mechanics with Einstein's theory of special relativity. And he ended up predicting these things called negative energy states for electrons.
Negative energy states.
Yeah, at first it seemed like just a weird mathematical thing, but Dirac had this amazing insight.
What was it?
He said that those negative energy states could actually represent a brand new particle, one that was just like an electron, but with the opposite charge.
So a kind of mirror image of the electron.
Exactly. He called it an anti-electron, and it was later renamed the positron.
Wow, and this was all just from an equation.
That's the power of theoretical physics. And then later on, they actually found experimental proof that antimatter exists.
Incredible. Okay, let's move on to NBL 443.pdf, which is Thomas Hunt Morgan's lecture on chromosomes and heredity. Can you remind us about chromosomes? What role do they play in genetics?
Chromosomes are these structures you find inside the nucleus of a cell. They're made of DNA, which is basically the instruction manual for building an organism.
Right.
Morgan used fruit flies to study how traits are passed down through generations.
Fruit flies, they seem kind of random.
They're actually perfect for genetics research. They reproduce really fast, they have a relatively simple genome, and their traits are easy to see.
Oh, I get it.
And Morgan was able to show that genes, which are like the individual instructions in that DNA manual, are located on chromosomes.
So he was mapping out the blueprint of life.
Exactly. He also discovered something called linkage, which is where genes that are close together on a chromosome tend to be inherited together.
That's fascinating. Okay, well, let's jump to NBL 444.pdf, which features Frederic Joliot-Curie's lecture. It's about artificial radioactivity.
The Joliot-Curies, they were pioneers in nuclear chemistry. They discovered that you could actually create new radioactive isotopes by bombarding stable elements with alpha particles.
Okay, can you back up a second? What's an isotope?
So isotopes are atoms of the same element that have the same number of protons but different numbers of neutrons. Some isotopes are stable, but others are radioactive, meaning they decay over time and release radiation.
Right, okay.
And the Joliot-Curies found that they could make stable elements radioactive by hitting them with alpha particles, which are basically helium nuclei.
So they were changing one element into another, kind of like alchemists.
That's a good analogy. And this discovery had huge implications. It led to the development of new medical treatments, new ways to do research, and even new energy sources.
It's amazing how one discovery can have such a big ripple effect. Okay, ready for our last Nobel lecture.
Let's do it. This one, NBL446.pdf, is George Beadle's lecture. And he's asking a really fundamental question. How do genes control the traits of a living thing?
A big question.
He and another scientist, Edward Tatum, used bread mold to study this.
Bread mold. Why bread mold?
It's easy to grow in the lab, and it has a pretty simple life cycle. So Beadle and Tatum used X-rays to cause mutations in the mold's DNA.
Okay, so they were messing with its genes.
In a way, yeah. And they found that certain mutations made the mold unable to make certain nutrients. And that meant the mold couldn't grow unless you added those nutrients to its food.
Interesting. So how did that help them understand how genes work?
Well, they realized that each mutation was affecting a specific enzyme.
An enzyme.
Yeah, those are proteins that help speed up chemical reactions in cells. And their work led to this idea, the one gene, one enzyme hypothesis.
So one gene makes one enzyme. I like it. It's simple.
It is. Of course, it turned out to be a little more complicated than that, but it was a huge step forward in understanding how genes actually control what goes on inside a cell.
Wow. Well, this deep dive has been absolutely incredible. We've covered so much, from the vastness of space to the tiniest details of life. I'm feeling inspired by all these scientists and their amazing discoveries.
Me too. It's a reminder that we're always learning and exploring, and there's always more to discover.
Well said. Until next time, keep exploring, keep asking questions, and never stop wondering.